visiting this morning that I've flown through Salt Lake City a million times, and I think this is the first time I've ever actually left the airport. So it was lovely to actually get to stay and spend the day here. It's beautiful and the weather is lovely. So today I'm going to talk a little bit about cardiac electrophysiology, but I'm actually going to talk a little bit more about cardiac signaling and some imaging approaches that we're using in our lab as well. Maybe. Okay, there we go. So for many years, my lab has really been interested in autonomic control of the heart with a particular emphasis on sympathetic signaling. And this is just kind of a refresher for maybe those of you who don't think about the nerves in the heart all the time. The heart is actually really rich with innervation. This is just some images of the sympathetic innervation. Let's see, yeah. Here, if you can kind of see that, that's a mouse heart with the sympathetic nerve stained in green. And both the atria and ventricles are really densely innervated by the sympathetic innervation. And recent studies have shown that the nerve to cardiomyocyte ratio is actually fairly similar to the capillary to cardiomyocyte ratio. And what that means is they're really dense. And every single myocyte in the heart is in contact with at least one sympathetic nerve, if not more. But despite this sort of rich innervation, the density is non-uniform. So for both sympathetic and parasympathetic innervation, there are gradients from the base to the apex. So the top of the heart has more nerves than the bottom. And that probably makes sense because the nerves sort of come in on the great vessels and sort of then project down toward the apex of the heart. And for the sympathetic innervation, there's a gradient from the epicardium to the endocardium. So there's seems to be denser innervation sort of on the outside of the heart, and it gets a little bit more sparse as you go into the endocardium. And over the years, it's been discovered that even in structurally normal hearts that have no underlying cardiovascular disease or structural disease, any disruptions to these nerve gradients seem to provoke cardiac arrhythmias. So that's what's shown here. It's from a mouse model that was transgenic for a neurochemorepellent that interfered with the normal patterning of the nerves across the ventricle. Nothing else was wrong with these mouse hearts, and they experienced sustained ventricular tachycardia. So this sort of implies that there's kind of a close communication between the cardiomyocytes and the nerves, and any disruption to that sort of normal communication can be arrhythmogenic. And so we know that a lot of remodeling happens to the sympathetic nervous system in cardiovascular disease. And when I say the word remodeling, I use that term very broadly, and that can include changes to sympathetic tone or just the overall sympathetic drive, the function of the nerves, the anatomical density and distribution of the nerve fibers throughout the heart, and even the neurotransmitters that the nerves release and the neuropeptides they release. All of these things can change in different forms of cardiovascular disease. And nearly all of these changes are in some way associated with cardiac arrhythmias. So again, any sort of disruption to this normal sympathetic signaling appears to be pro-arrhythmic. And over the years, my lab has sort of looked at this in a variety of different models and a variety of different pathologies and disease states, including aging, different forms of heart failure, what happens after animals are exposed to nicotine or secondhand smoke. This also disrupts sympathetic signaling. And we've spent a lot of time over the years studying myocardial infarction, and that's what I'm going to focus on today. So these images are not from my lab. They're from the group at UCLA, but I thought they were really beautiful images of a normal mouse heart with the sympathetic innervation in white, and then a mouse heart after myocardial infarction, so after a coronary ligation. And you can see that this area is largely devoid of nerves. And at the border zone, sort of this area near the infarct, you can see that the infarct itself is devoid of nerves, but in that border zone, there's lots of nerves. So it's almost hyperinnervated. So you can get these areas of hypoinnervation adjacent to areas of hyperinnervation. And these images here, where the color dictates the size of the nerve fibers, so you can see the structure and size of the nerve fibers is completely different than it is in the normal control heart. So there's a lot of remodeling of innervation that happens after myocardial infarction. But what is a little bit surprising is, if you think about it, so the infarct itself the cardiomyocytes die after the ischemic event or due to the ischemic event, but the nerves also die. And then as part of the healing process, a lot of the dead myocytes are cleared out. It's replaced with fibrosis, but there are quite a bit of surviving myocytes in this region. And what happens is the nerves sort of grow back, and then they grow up to the edge and they stop. But the sympathetic nerves, as part of the autonomic nervous system, it's peripheral nervous system. It's not central nervous system. So it should regenerate. But something is preventing those nerves from growing into the infarct. And over the past decade or so, together with one of my close collaborators at Oregon Health and Sciences University, Beth Habecker, we've found that there's expression of chondroitin sulfate proteoglycans, or CSPGs, as something present in the infarct. We still don't know what cell type is making it or why it's there, but it's there. And what happens is when the sympathetic nerves are regenerating and trying to grow back into the infarct, the CSPGs bind to a receptor on the sympathetic nerves that repels them and blocks them from growing in further. And that receptor is the protein tyrosine phosphatase receptor sigma. And so Beth Habecker, together with us in a few studies, we showed that this was prevalent in rodent hearts. And then recently, this last year, we also showed that in a rabbit model of myocardial infarction, the infarct is really robust in expression of these CSPGs. And basically, wherever there's CSPGs, there's almost no innervation. So the sympathetic innervation here is labeled with tyrosine hydroxylase. And you can see even in the border zone, wherever the CSPGs are, the nerves sort of grow up to the edge and then they really stop compared to the normal nerve distribution. So this seems to be preserved across species. It happens in rodents, it happens in larger animals. And Dr. Habecker has shown over the years that if you inhibit the PTPRS receptor on neurons, so that essentially the neurons don't see these CSPGs, then you can promote the nerves to grow right back into the infarct. And you can do this either genetically by knocking out that receptor in the neurons, or you can do it pharmacologically. And this is a study from a couple years ago from Dr. Habecker's group, where she used a couple of small molecules, as well as a peptide called intracellular sigma peptide. We treat the animals with these drugs after myocardial infarction. You can promote nerve growth back into the infarct. And importantly, that actually works to prevent cardiac arrhythmias. So when we first started these studies and Beth Habecker was talking to me about them, she said, but let's grow the nerves back into the infarct. I was like, why? That might do more harm than good. But it turns out that restoring that innervation, potentially by homogenizing the innervation, by reducing the heterogeneity, by restoring sort of that normal sympathetic activity, tends to reduce arrhythmias. And so that's shown here in mouse models treated with these different drugs to restore innervation. They have much fewer premature ventricular contractions after injection of isoproterenol. So many years ago, we decided to try to understand this more mechanistically. Why would this be preventing cardiac arrhythmias? So we did a study together with Dr. Habecker where we had our normal mouse model of infarction, where you have the scar and the nerves sort of growing up to the edge and stopping. And then we had a genetically modified mouse where the receptor, the PTPRS receptor in neurons, was knocked out, so the neurons could no longer sense the CSPGs. And the neurons, in this case, grow back into the infarct. And in my lab, we do a lot of optical mapping. So we take hearts, we excise them, perfuse them outside the body. We load them with voltage and calcium sensitive dyes, and then we look at the action potentials and the calcium transients to try to see if we can understand more deeply the mechanisms that might be contributing to arrhythmias. And so that's what we did in this study. And so these are action potentials in black and calcium transients in red from the control heart, which is sort of the infarct, the normal infarct. And you can see under control conditions, the action potential upstroke precedes the calcium transient upstroke. So this is normal excitation, contraction, coupling. And when we added a beta adrenergic agonist like isoproterenol, you can see we start to get calcium mishandling, particularly in the infarct zone where calcium is being released. it's being elevated intracellularly. And if that calcium, diastolic calcium rises enough, this can trigger an action potential. And so that's what we were seeing in these normal hearts. Whereas in the hearts that had that were re-innervated, you can see when we added isoproterenol, it was still normal excitation contraction coupling, and we weren't seeing this pathological calcium mishandling. And so what we concluded from that study is that this denervation, this loss of innervation, that the cardiomyocytes are normally used to seeing, this normal sympathetic adrenergic signaling that's happening all the time, once that's gone, what can happen is perhaps the myocytes are becoming super sensitive to catecholamines and super sensitive to adrenergic stimulation. So for what might be a normal physiological dose of catecholamines, it induces calcium mishandling and pathological activity in response to the catecholamines. So we started calling this denervation supersensitivity, and this has been shown in other models outside the heart. But it's very kind of a classical response that if innervation is lost, the tissue that's normally innervated can become super sensitive to catecholamines. And this is just some quantification of what we found. Again, with isoproterenol, we had lots of PVCs, or premature ventricular contractions, and they were prevented with a beta blocker, just showing that, again, this does seem to be due to this adrenergic receptor stimulation. And when we did our optical mapping, we were able to also take a look, and it seemed like these premature ventricular contractions were arising from that infarct area. So from that denervated area seemed to be very sensitive to catecholamines, calcium mishandling, and triggered activity. So this is some work that we did many years ago. And so we sort of concluded that it must be this phenomenon of denervation supersensitivity. But everything that we were doing sort of functionally to understand this process, we were really looking at what I'm calling sort of these downstream functional outputs. So by the time the nerve releases the catecholamine and binds to the adrenergic receptor, you have a whole intracellular signaling cascade that phosphorylates a bunch of ion channels and calcium handling targets. And then we're seeing the result of that, right? So this is a pretty downstream kind of end functional data. And so over the years, we really were thinking deeply about this question, and we were wondering, is there a better way or another way that we can assess how these hearts are responding to adrenergic activity? So if we look at this little diagram, this is just the end of a cartoon of a sympathetic neuron and a cardiomyocyte. And normally the norepinephrine is released from the sympathetic neuron. It binds to beta adrenergic receptors that activates adenylyl cyclase to make the second messenger cyclic AMP. Cyclic AMP then goes on to activate protein kinase A. Protein kinase A goes on to phosphorylate a bunch of downstream targets. And here's those action potential and calcium transient results that we were seeing before. And just as a note, because we'll get to it later, cyclic AMP, to turn this signal off, cyclic AMP is degraded by these things called phosphodiesterases, or PDEs. But we were looking at this and we were thinking that maybe cyclic AMP would be a good sort of intermediate second messenger that we could take a look at to understand how the heart is responding to adrenergic stimulation. So can we look at this second messenger before we sort of see the downstream functional responses? And so this type of work has been done quite a bit in different systems, particularly with the use of biosensors, including FRET sensors. So there's many different FRET sensors for cyclic AMP that bind cyclic AMP, and that can change the position of a CFP and YFP fluorophore, and you can record that as a change in FRET. And so this has been done a lot in isolated cardiomyocytes where the hearts are isolated, the cells are digested. You can either then express one of these fluorophores and study how the heart responds to adrenergic activity at the cellular level. So that's nothing new, that's been done a lot. But we're really interested in arrhythmia mechanisms and what's happening in the infarct and the border zone and the remote zone and how this might be contributing to arrhythmias. So we were really interested in doing this at the whole heart level. So to do this, several years ago, we came across, we didn't make the mouse already existed, called a camper mouse. So it expresses an EPAC-based cyclic AMP FRET sensor. But we were able to cross that with the alpha-myosin heavy chain Cree to get cardiac specific expression of this cyclic AMP biosensor. So this mouse had been used in neuroscience applications, and I think had been used to study insulin signaling, but had not yet been used to study the heart. And then what I'll get to in a moment is through much trial and error and many iterations, we were able to adapt one of our optical mapping systems to do this imaging in the intact heart so that we could see in real time how the heart was responding to adrenergic activity in terms of generation and breakdown of cyclic AMP, this key second messenger. So to do this, we started with one of our optical mapping systems, which was already built to record calcium and voltage sensitive dyes through 2 detectors. And we were able to sort of modify the system and add a beam splitter and basically split all of our wavelengths well enough to also record the CFP and YFP of the fret sensor. So we're able to detect 4 different wavelengths with this setup. This was essentially ended up being the projects of a very talented postdoc, Jess Caldwell, as well as a bioengineering PhD student, Eric Lee. And so we built a couple of different of these systems. This is the kind of a photograph of what our current most optimized system looks like now. And we're calling it the CAMP FRET plus dual optical mapping. So the acronym that we came up with was FREEDOM. And the first time we got this new system sort of built and up and running and flipped the lights on, Eric and Jess had to come run and get me in my office and bring me in because they wanted to show me something. I was like, what do you need to show me? And they said, it's freedom. You need to see freedom. And I was like, okay. And they're like, freedom casts the shadow of an eagle on the wall. I was like, I think that that's a stretch, but okay. So anyway, so this is freedom. So when we add, so we started out just doing very basic experiments. We took these sort of normal hearts expressing this cyclic AMP sensor, and we started just doing Langendorf perfusion and then just doing small little boluses of norepinephrine. So just the normal neurotransmitter that should activate adrenergic signaling, wash the norepinephrine in and out. And, you know, we could record our changes in CFP and YFP as we expected. We can then go through and calculate the fret ratio on a pixel by pixel basis. So we can do this in 2 dimensions. And then because we've integrated this with our optical mapping, we're basically what we're doing is we're doing time-lapse fret imaging. So as we're doing sort of our norepinephrine application and watch out, we can look at the cyclic AMP signaling, let's say every 5 or 10 seconds. We can sort of, because that seems to be a slower process of cyclic AMP generation and breakdown. And that kind of makes sense, that sort of time scale, because you think if you get scared or you get startled by something, you feel that, oh, didn't mean to bang on that. You feel your heart racing a little bit, and then it sort of, you know, dissipates over, 30 seconds, a minute. So this is sort of a longer time scale where we're sampling every 5 or 10 seconds to look at these cyclic AMP kinetics. But if we want to resolve the mouse cardiac action potential and calcium transients, we have to record that at very high speed. So after every single fret frame, then we flip on our optical mapping cameras and we record 1 or 2 seconds of data at 1 kilohertz speed so that we can actually then resolve the calcium and action potential. So these colored dots here indicate action potentials that are recorded at these different stages of our protocol. And then at each location on the heart, we can go back and we can say, okay, at this, area of tissue, at that moment, what was the exact magnitude of the cyclic AMP signal, and what did the calcium transients and the action potentials look like at that location at that moment in time? So we can start to sort of put together how the second messenger is then driving the functional outputs. So our first studies, we just took normal hearts, no disease model. We just took these camper hearts, and we took some males and females, and we challenged them with norepinephrine. So we did this bolus of norepinephrine, wash it in, wash it out. And I had this very sort of naive thought that if we just wash in norepinephrine and wash it out, the cyclic AMP should just rise uniformly and then degrade uniformly everywhere, right? And that's kind of what we saw in the male hearts. So this is this time-lapse cyclic AMP fret. You can see it lights up everywhere and then decays. And if we take regions of interest from the right ventricular base and the apex and compare them, that's what's plotted over here. And you can see the apex decays maybe a little faster, but it's mostly uniform. Whereas what we started to see in the female hearts was that everything sort of rose a little bit more uniformly. But during the decay phase in the females, the apex signal, the cyclic AMP signal in the apex always degraded faster than it did at the base of the heart. And that's what's shown here. And like I said, that trend was kind of there in the males, but it was very pronounced and significant in the females. And so this is just a movie of that. So there you can see they both light up. And then it's slowed down here in time. But you can see the females, they kind of decay from the apex back up to the base. I think I can show it one more time. And it kind of happens in the males, but it was much more pronounced in the females. And so then we went back and we looked at our voltage data, and we looked at the action potential properties at every location over time. And we plotted these two-dimensional maps where the color represents how did the action potential duration change? Because cyclic AMP can turn on, for example, L-type calcium currents in the mouse heart, make the action potential longer. And then as those currents, as the cyclic AMP is decayed, those currents go back to normal. So we can measure these precise changes in the action potential duration. And I think this is from sort of this time point here during the cyclic AMP decay phase. So you can see in the male hearts, when the cyclic AMP has sort of uniformly decayed everywhere, we see that this map is mostly blue, meaning that the action potential has sort of shortened uniformly everywhere. Whereas in the female hearts, you can see this the heterogeneity in cyclic AMP led to quite a bit of heterogeneity in the change in the action potential duration. So up here where the cyclic AMP signal is still high, the action potentials are long because there's lots of L-type calcium current still flowing and the action potentials are long. Whereas down here in the apex where the cyclic AMP signal has degraded, the action potentials have shortened up. So we're getting these really heterogeneous changes in electrophysiology. And we found, I didn't have the data here, but we found that that could actually change the repolarization sequence in the females, but not the males. So it really did lead to abrupt differences in the repolarization sequence in the females, but not the males. So we did a lot more work on this and tried to investigate underlying mechanisms. And I'm gonna jump over some things and basically tell you that what we discovered is we think that these differences are due to the changes in expression of phosphodiesterases or PDEs. So remember, that's the enzyme that breaks down the cyclic AMP. We found that the total PDE activity was elevated in the females compared to the males, but particularly at the female apex, both compared to the female base and compared to the males. So there seems to be more of that enzyme in the apex of the female hearts that's quickly degrading the cyclic AMP. It's leading to action potential changes. It's leading to changes in repolarization, which at this point, we don't know if this would necessarily be pro or antiarrhythmic in the females. So these are just normal mouse hearts. They usually don't have a lot of arrhythmias, but it essentially allowed us to begin to sort of understand these mechanisms by which cyclic AMP might be broken down differentially at different parts of the female heart and differently between males and females. But our whole goal of when I sort of started this talk was to look at myocardial infarction, right? So those were just normal hearts, males and females. But then we wanted to apply this approach to myocardial infarction. So again, this is just to remind you of the model. And in this case, this is just showing that data from 2015 again. In that published data, this was a genetically modified mouse where the receptor to CSPGs in the neurons were knocked out. However, for this upcoming study, we instead chose to use pharmacological approaches. So we took our camper mouse hearts that express the fluorophore. We created myocardial infarction. We waited 3 days. And then from days 3 to 10, we treated the animals with ISP, or intracellular sigma peptide. So that basically binds to the PTPRS receptor in the neurons, so the receptors don't see the CSPGs, and the neurons can grow back in. So we did that, and then we basically repeated these same protocols where we were doing our cyclic AMP imaging, and applying norepinephrine. So when we did that, so here's our sham hearts, and you can see, this average cyclic AMP trace, just the increase in decay, similar to what we saw in the normal hearts. And what we found was in the MI hearts, the cyclic AMP generation was actually quite a bit reduced, but also this decay kinetics were completely altered. So now we no longer have this rapid decay. We had this very prolonged decay, and you can even sort of see that here in this image compared to the sham image. And so this was really surprising to me because remember I said, back from 10 years ago, we thought that denervation after MI would make the heart super sensitive to catecholamine, so that they were having this super response to a normal dose of catecholamines, and that's what led to arrhythmias. And in my mind, a super sensitive response meant more cyclic AMP, right? They might just be generating tons of second messenger, which is then causing this whole downstream cascade of events. And that's not what we saw. We actually saw it was a little bit less cyclic AMP, but the kinetics were totally different. So there's less being generated, but it might be sticking around for too long, right? So it's not being broken down like it should. And these are just the decay kinetics shown here, that it's significantly prolonged after myocardial infarction. And what we attribute that to, well, I think we'll get to that here. And what we found is when we saw the premature ventricular contractions in response to norepinephrine, they typically occurred during these later phases where cyclic AMP should have been degraded and wasn't. And then when we also went in and stimulated, when we bypassed the receptors and we just stimulated adenylyl cyclase with a drug called forskolin, again, we saw lots more PVCs in the MI hearts when we generated cyclic AMP, suggesting that cyclic AMP signaling is in some way impaired. Again, when we did our detailed optical mapping, we saw that the PVCs were arising sort of from this infarct area or sort of adjacent to that infarct area. Again, the area that we think is denervated and is super sensitive. And then we went on and we treated our animals with the drug to grow the nerves back, right? So again, we gave these daily injections of ISP, and lo and behold, it didn't really restore the peak amplitude of the cyclic AMP, but it really changed these decay kinetics again. So now all of a sudden, we're still making a little bit less cyclic AMP than before, but we're getting this rapid, robust, efficient degradation. And that was reflected in this time constant of the decay here. So you can see the drug reverse that. And again, now when we looked for PVCs, there were no PVCs in response to norepinephrine out here at these later stages. And when we used the drug forskolin, again, no PVCs in that treated group. So something about growing the nerves back is contributing to cyclic AMP degradation and turning that signal off. And that was not at all what we expected to find. And so this, of course, got us wondering, well, why would that be happening? What could possibly be the mechanisms at play here? So if we go back to this little diagram, again, we see that norepinephrine is released from the sympathetic nerve terminal, binds to beta adrenergic receptors that activates adenylyl cyclase to make cyclic AMP. And then the cyclic AMP is made right here by the receptor, but also near the beta adrenergic receptor, there's an anchoring protein called SAP97, which holds this particular PDE, PDE4D, very close to the receptor. And we think that that sort of localization of PDE4D here is really important for sort of rapid and efficient degradation of the second messenger and kind of turning off of that signal. And also, not only turning it off rapidly, but then preventing this cyclic AMP from diffusing to other locations in the cell and signaling throughout the cell where maybe it's not supposed to. So why would nerves matter? This is all happening within the cardiomyocyte. Why would having the nerve here matter? And so we dug through the literature, and we found several years ago a study that was published from Brian Kobilka's group, where they did co-cultures of cardiomyocytes with and without sympathetic neurons in the co-culture. And what they found is that when they co-cultured the cardiac neurons with the sympathetic nerves, and so that's what's shown here. This is, I think this is a big cardiomyocyte here. And then this is the nerve sort of lying across the cardiomyocyte. When you zoom in and take a look at that, they found that within the myocyte, the expression of this protein SAP97, this one, lined up perfectly underneath the nerves. So expression of this protein seemed to be preferentially aligned within the cardiomyocyte wherever there was a nerve. So there's something structurally or functionally or chemically about having that nerve there that the adjacent cardiomyocyte senses that, knows that, and we think maybe more appropriately sort of constructs these adrenergic signaling units to be right there at the nerve where the neurotransmitter is going to be released. And then our hypothesis is what might be happening after myocardial infarction then is if you lose that neuron, the cardiomyocyte senses that somehow. We don't yet know how that happens, but there's no longer the robust expression of this anchoring protein. So the PDEs are not being held right at the receptor. You might have too much cyclic AMP and then activation of PKA. And there's been some other reports, including by Kevin Zhang in our group or in our department at UC Davis, showing that when you lose SAP97, that could also lead to abnormal pathological CAM kinase activation and sort of some other cellular signaling events that may predispose to arrhythmias. So this is sort of our working hypothesis of what might be happening. So we then went on and we said, okay, well, can we look at PDE activity functionally a little bit rather than just looking at those decay kinetics? Are there other ways to look at this? So we did an experiment, again, same 3 groups of heart, sham, myocardial infarction, and myocardial infarction with the drug to grow the nerves. And we first added the drug forskolin. So we bypassed the receptors. Forskolin just activates adenylyl cyclase to make cyclic AMP. And see everything lights up. We're making cyclic AMP. And if we look over time, that's sort of this first bump where we see the signal increase. And then we added a second drug, a PDE inhibitor, IBMX. So now we're inhibiting the breakdown of cyclic AMP with this drug IBMX. And you can see then we get a second sort of little bump in the signal. And that we think that that change in signal when we add IBMX is reflecting how much sort of baseline cyclic AMP breakdown by PDEs is going on. Because if the PDEs are all active here at this steady state, as soon as you inhibit them, the signal jumps up, and that gives us some indication of how much work those PDEs were doing under normal circumstances. And so when we calculate the ratio here and look at that, again, we saw that after myocardial infarction, the PDE activity was decreased. And then when we added this drug to regrow the nerves, that activity appears to be elevated again. So there's some more functional PDE activity. So we think that that's a little bit consistent with our hypothesis. But also, we found that PDE expression alone doesn't seem to reflect this. So if we had reduced PDE activity after MI and those reduced decay kinetics after MI, we might think that PDE expression should be decreased after MI. That's not what we found. So we looked at a couple of the major PDE isoforms in the mouse heart. And in MI, they definitely weren't down, and they might have been even a little bit elevated. And then when we added the ISP, it either kind of stayed the same or maybe elevated just a little bit more. So it doesn't mean that after MI, the heart is lacking phosphodiesterases. It seems to have plenty of them, but maybe they're not in the right place. Maybe they're not being held at the receptor to perform the function that they need to, which is then leading to sort of the cyclic AMP signaling being prolonged and being moving throughout the cell in ways that are pathologic and promoting arrhythmias. So this is very much a work in progress, and now we're working on trying to work with some collaborators and do some more biochemistry experiments to do some co-IPs to look at these complexes and see what's pulling down together so that we can really nail down, is it the loss of this anchoring protein that's going on here, and does reinnervation help restore this anchoring protein to restore this signaling complex? So that's very much a work in progress. But so far, everything's kind of consistent with our cartoon here. And then just sort of one other kind of new thing that we're working on now that I just wanted to show you real briefly is another reason why that cyclic AMP signal might be prolonged with nerve loss is that the nerves are actually really important for removing neurotransmitters from the receptor, right? So around 90% of the neurotransmitter that is released by the cardiac sympathetic neurons is taken right back up by that neuron, right? So they take it up, they recycle it, they release it again. So then having the nerves there is pretty important for neurotransmitter clearance. And so another contributing factor that we think might be going on here is that when we don't have this nerve here, and there's an uptake transporter called the norepinephrine transporter, if that's gone, the nerve is gone, none of this norepinephrine is being rapidly removed. It's maybe just stimulating the receptor for too long, right? And that would be another contributing mechanism. If there's just too much catecholamine here, continuing to stimulate the receptor, regardless of what's happening to the PDEs, that you're just going to keep making more cyclic AMP for longer. And so, again, we asked, because I'm sort of from an engineering background, and is there a better way that we can measure this? So up to this point in the talk, everything I was showing you was looking at cyclic AMP signaling with a biosensor here. We asked, can we go upstream, and can we look at the norepinephrine signaling in the cleft here? And can we do that with another biosensor? So there's another family of biosensors called G protein coupled receptor activated biosensors, GRAB sensors. And so there's a GRAB sensor that can sense norepinephrine. So it's just based on it's a non-functional alpha adrenergic receptor that has a circularly permuted GFP on it. When that adrenergic receptor isn't bound to anything, the fluorescence is low. As soon as the alpha adrenergic receptor binds either norepinephrine or another alpha adrenergic agonist, then that causes this robust signal expression. So again, we didn't create this mouse. This mouse existed, again, from neuroscience applications. The neuroscientists have beat us to it many times. But we were able to take that mouse and cross it with the alpha-myosin heavy chain to get cardiomyocyte specific expression of the GRAB sensor. So the first thing that we did was we just took these hearts and we perfused them. And then we just put on increasing doses of norepinephrine just to show, does our sensor work? Turns out it does. We can see this fluorescence increase proportionally with increasing doses of norepinephrine. The heart lights up beautifully, so the sensor appears to have a nice dynamic range. And so then we went back, and instead of doing the simple, easy experiments, I said, let's do some MIs. Why not? Just go to the disease model, see what happens. And so we then wanted to look and see, can this GRAB sensor report on norepinephrine signaling after myocardial infarction? And so one thing I kind of didn't tell you at the beginning of our talk is everything I presented so far up to this point today, we've just been exogenously applying catecholamines to the bath. So just giving a little shot of norepinephrine into the perfusion. Our lab over the years has developed methods to stimulate catecholamine release other ways, including isolating hearts with the spinal cord and the sympathetic nerves intact. So that's what's shown here. This is a mouse heart. This is the back of the rib cage. The spinal cord is kind of hiding here behind this is our perfusion cannula, perfusing the descending aorta. And if we take an electrophysiology catheter and we put it up the spinal cord to about the first or second thoracic vertebra, and you stimulate electrically, you can stimulate all those sympathetic projections that are coming off to the heart and stimulate the heart's own nerve release. So that's what we did here. Again, sorry, I jumped about 10 steps, but I just wanted to show you some cool preliminary data. So we took one of these GRAB hearts that had myocardial infarction, and this time we stimulated the nerves and we imaged. And you can see when we did the sympathetic nerve stimulation, or SNS, now you can see everything in the heart lights up except that infarct zone, right? So the infarct zone is denervated, so it stays really dark. But the nerves are releasing catecholamines all around the rest of the ventricle, and you can even see the atria get really bright because the atria are also really densely innervated. So we're able to sort of use this as a relative readout of our nerve release catecholamines. So that's what's shown here. And then, so this is as we would expect, infarct zone is dark. Then we went back and we did the opposite experiment and we gave a little shot of norepinephrine. Okay, now what if we just give some norepinephrine? And this I was not expecting to see, but I thought was super cool. So we gave norepinephrine, and again, the heart lights up everywhere. So now even the infarct lights up. So it's getting norepinephrine. There's lots of surviving cardiomyocytes here. They're all expressing that grab sensor. They're lighting up. But now look what happens during the sort of that norepinephrine washout phase. So when we apply exogenous norepinephrine, we think a lot of it's probably just being washed out in the bath. But some of it's being rapidly removed from the cleft by the nerves. And you can see that here because where all the nerves are up here, that signal rapidly decays. So the nerves are taking up all that norepinephrine, removing it as soon as it's unbound from the receptor. Whereas in that infarct area, it's lingering because there's no nerves to help take away that norepinephrine. So that signal might be lingering for longer. And that's what's shown here. And so we think that that's probably just another contributing mechanism that not only maybe is the phosphodiesterase activity reduced, the second messenger's sticking around too long, maybe the neurotransmitter right at the receptor is sticking around too long too, because we don't have those nerves there to efficiently remove neurotransmitter. And so this is kind of a brand new area that we just started working on. So this was very new data that I just wanted to show for fun. So this is, we think that this denervation probably contributes to disrupted clearance of norepinephrine at the neurocardiac junction. And that's very much a work in progress to be continued. So I will end there with just a few conclusions. So we think that our methods for cyclic AMP imaging have shown that there's region and sex-dependent differences, particularly in phosphodiesterase activity. And this has kind of led me, at least, to have a shift in my thinking, because I've thought about autonomic control of the heart for a very long time. But I've really only ever thought about it from the standpoint of turning on of the signal, whether that's where the nerves are, how much norepinephrine they're releasing, where the receptors are, how the receptors are responding. I had not, until we really started working on these projects, given much thought to the turning off of that signal, maybe just as important or more important than turning on. And so we're kind of changing the way that we think about this. Oops. And then we found that in normal female hearts, they have increased PDE activity at the apex. And like I said, this may or may not have implications for arrhythmogenesis. So those studies were all in normal mouse hearts that don't have a lot of arrhythmias. I could sort of make a theoretical argument how it could both be pro or anti-arrhythmic, depending on the situation. I will say so far in our MI models, we're not seeing huge sex differences. We are seeing some sex differences. They are at the apex, but they're very subtle. And that could just be because the MI itself is such a kind of stressful pathological insult that the pathological phenotype is so strong that it's sort of overpowering, and maybe these kind of more subtle sex differences are not as important in the pathological setting, but we're continuing to investigate that. And we think that PDE activity is somehow reduced after MI, and we think that restoring sympathetic innervation can help improve PDE activity and cyclic AMP signaling. And now we're working to determine whether it's localization, what PDE isoforms are involved, what anchoring proteins may be involved. And there's some good preliminary data that sort of suggests that we might be on the right path to something there. And also in our lab, I didn't talk about it today. Today, I just really kind of focused on the normal hearts and the MI. But now we kind of have this set of tools that we've developed over the past several years. So now we're applying this to a lot of different situations, including physiological nerve activity. So we're doing more studies where we're actually stimulating the nerves at the spinal cord that I talked about. We have separate studies where we're looking at the sinoatrial node and the atria and looking at cyclic AMP signaling at the level of the atria. And we also have a whole other project on non-ischemic heart failure. So this is ischemic MI. We're also working in a TAC model and trying to understand what happens in non-ischemic heart failure. And we think that imaging of this grab norepinephrine sensor can help provide us with sort of another link in that sort of whole adrenergic signaling cascade, and we can begin to keep probing different levels of this signaling cascade. And so I will end there, hopefully with a few minutes for questions, and thank everybody in my group and our funding. Most of the work I presented today was done by a very talented postdoc, Jess Caldwell, who is in the market for a faculty position. She has a K99R00. And Eric Lee, who is about to graduate with his PhD in biomedical engineering, and he really helped with a lot of the technical aspects. So, thank you.